Well, thanks very much for the opportunity to come and talk to you this morning. And what I want to do is to describe some of the work that we've been doing in the lab recently. Some of this work does not, I'm afraid, involve Arabidopsis, but that's a sign of the times. So I'm going to start off with a little bit of introduction and background for those of you who aren't hardcore epigeneticists. And then, as I say, I'll go into our more recent data. So I'm going to be talking about RNA silencing. So this is a process involving small interfering RNAs. And the particular variant of the RNA silencing pathways that I'm going to be discussing in my talk this morning is one in which the end result, rather than being a post-transcriptional effect at the RNA level, is an effect at the chromatin level. So this process, as is always the case, starts with DNA. That is transcribed into RNA. And the polymerase that we think is involved in the transcription process is a variant form of polymerase II. So some of the subunits in red are shared with polymerase II, and others are specific to polymerase IV. So this is a variant form of polymerase II, which generates single-stranded RNA. The single-stranded RNA is then converted by an RNA-dependent RNA polymerase into a double-stranded form. And the double-stranded form is then processed by an enzyme, an RNase 3-like enzyme known as dicer, into the small interfering RNAs. And in this particular variant pathway, the dicer generates small interfering RNAs that are 24 nucleotides long, different from the 21 nucleotides, which is characteristic of microRNAs and the small interfering RNAs associated with post-transcriptional regulation. So, having produced the small RNAs, what happens is that they then associate with Argonaut proteins. And there are a whole family of Argonaut proteins in Arabidopsis. So 3 in particular have been implicated in the type of pathway that I'm discussing this morning, associated with epigenetic modification. So having associated with the Argonaut proteins, the small interfering RNAs are then able to target introduction of epigenetic marks, so methylation in this particular example of the DNA, at the locus from which the whole process was initiated. Now a question arises then, how is it that RNA can target a protein complex to DNA? Well, we don't think that it does this by binding to DNA. This is work from Craig Picard's lab. What we think it does is that it interacts with a nascent transcript, a transcript that is still associated with the DNA, a transcript that is, in many instances, although not all, generated by another variant form of polymerase II, and this we know as polymerase V. In some instances, we think that polymerase II may be involved, but that's a sort of detail for specialists in this field. So the targeting event is to a nascent transcript. Now, having initiated then this first round of this epigenetic process, we think in some instances what happens is that the methylation of the DNA actually predisposes further transcription by polymerase IV so that you reinforce this whole process. And then once the small interfering RNAs are being produced abundantly, this then provides the opportunity for the Argonaut complexes to find similar sequences elsewhere in the genome, so that this process can act not only in cis, but it can also act in trans. The limiting factor being, or amongst the limiting factors, being sequence similarity or complementarity between the small interfering RNA and the target process. So this is number one bit of the background that I wanted to describe to you, and many labs have contributed towards our understanding of that. I've mentioned the Picard lab, I'd mentioned Thierry Lagrange and Steve Jacobson and various others, Marjorie Matsky, who've contributed to that, as well as my own lab. The second bit of background that I would like to describe to you is just the link between RNA silencing and a mobile signal. Now, this was first described by Olivier Voinet when he was a PhD student in the lab, now some years ago. And Olivier did a whole series of experiments where he initiated RNA silencing by transient expression of a GFP transgene in a leaf of a GFP transgenic plant. And what he observed was that the silencing effect spread from this leaf through the plant so that eventually you had systemic silencing. We believe the signal of silencing is moving through plasmodesmata as well as through the phloem. We think it's symplastic movement. And associated with this process, there's an amplification capacity. So what we know is that once you've initiated this whole process, you can remove the leaf in which the RNA silencing is initiated. But even so, as long as you've left it on for a day or two, the silencing will persist in the plant. So the signal moves out of this leaf, and then its effects keep on going. So presumably, it's able to initiate secondary siRNA production once the whole process is started. When this was first discovered, this mobile signal of RNA silencing, we could hypothesize that the signal was actually an RNA molecule, a novel concept at the time, because we hadn't really thought of RNAs as being mobile molecules in a plant, able to move long distances. But we didn't, at the time, know what was the identity of this signal. It presumably was signal that was related to the target of transgene, but in principle it could be long double-stranded RNA, it could be short RNA, it could be any of several things in between. And so, and we also thought, when we first did these experiments, that the mobile silencing was associated with post-transcriptional silencing rather than the chromatin directed process that I've just described to you. As I'll show you from our recent data, we now know definitively what the signal molecule is, and we now know that this distinction between post-transcriptional and transcriptional silencing in the signaling process is actually not valid. So the approach that we used in order to investigate the signal molecule involved grafting. So the idea is that you take one genotype of plant and you graft it to a bit of another genotype of plant, and then you look in the other genotype, and if you see RNA that is identical to the genotype that you put in the first part, then the RNA must have moved across the graft union. It must be the mobile RNA. Now, we tried these types of experiments a long time ago when Olivier first discovered the signal of silencing, but because background and so on and so forth, we were not able to detect the mobile RNA. There was always a background signal, and you could never convince yourself that you were looking at something that had actually moved. But what we're able to do now is to use Illumina sequencing as a highly sensitive way with a very large dynamic range, very sensitive of looking for molecules. And we do these experiments in Arabidopsis, and the way we do them, because these signals move from photosynthetic source to photosynthetic sink, and because Arabidopsis doesn't have stems that are easy to graft or vegetative stems, we have monitored the movement of the signal going from the shoot to the root of the plant. So you've got different genotypes of plant. Some of them are mutant for the RNA silencing pathways. And then you've looked at RNA going from this part of the plant into this part of the plant. Now, having done Illumina sequencing, we've got a genome-wide profile of what these mobile RNAs are. But in order to make what I'm telling you easier to understand, I'm just going to describe what is happening at a couple of loci. And the locus that I'm going to describe to you first of all is the pi locus. So pi is an enzyme involved in tryptophan biosynthesis. Judith Bender did some work on the system a number of years ago, and she described how in different genotypes of Arabidopsis there is a sequence polymorphism at one of the pi loci. So in the C24 genotype, you've got an inverted repeat, whereas in Columbia, you haven't. What this means is that you can get read-through transcripts in C24 that fold back on themselves to get a double-stranded structure that can be processed by DICER to generate small RNAs. So what you would expect is that C24 produces small RNAs from pi, whereas Columbia doesn't. So you can then ask whether or not pi-specific small RNAs move from C24 into Columbia. So this is the control, and there's nothing there. And that's what we wanted to see because it's an extract of Columbia roots, and there are no small RNAs corresponding to the pi locus in the Columbia roots. If we look in the C24 shoots, we're looking along the pi locus here, and this is the number of small RNA reads that we see. We can see there are several all the way along the pi locus. And we also when we look at grafted Columbia roots, grafted to a C24 shoot, we now find indeed that we do get pi-specific small RNAs. So what we can conclude from this experiment is that pi-specific RNA must have moved from the top of the plant to the bottom. Now, from this experiment, we can't necessarily conclude that they've moved as small RNA. They could have moved as a precursor and have been processed in the lower part of the plant. And I'll address that in the experiment I'll describe to you in a moment. I just want to point out that there's a bit of additional information in this slide because we've color-coded the RNAs according to their size. Red ones are 21 nucleotides long, whereas blue ones are 24 nucleotides long. And you remember the distinction, I hope, associated with the post-transcriptional and transcriptional silencing or chromatin silencing pathways. In the root grafted onto this shoot, we only see blue RNAs. And so it looks as if at this locus in particular, there is selectivity for movement of the 24 nucleotide RNAs or precursors. To address whether or not it's the RNA or the precursor, the small RNA or the precursor, we did the same experiment, but using a Columbia genotype that is mutant for dicer 3. So this is the dicer that is not able to that generates 24 nucleotide RNAs. The dicer 3 mutant is null for this protein, and so it doesn't generate its own small RNAs, but nevertheless, its own 24 nucleotide small RNAs, but nevertheless, we see abundant 24 nucleotide small RNAs in the root. So small RNAs, not the precursor, the small RNAs themselves have moved from the root to the shoot. This is an experiment in which we used as the shoot a polymerase 4 mutant of C24. So this is not able to generate abundant 24 nucleotide small RNAs. It has predominantly 21s from the pi locus. And now we don't see any RNA that has moved down. So this reinforces the idea, at least at this locus, that there is selectivity for 24 nucleotide RNAs in the mobility mechanism. The next question then that arises is, having demonstrated that small RNAs can move, these are endogenous small RNAs, and I should emphasize that there are many thousands of loci within the Arabidopsis genome, most of them are transposon related, that have the potential to generate these mobile small RNAs, and many of them do generate mobile small RNAs. Having asked whether or not these RNAs can move, we then wanted to confirm that these endogenous RNAs could actually do something where they got to where they're going. And in this 24 nucleotide pathway, as I've described to you, one of the end results is DNA methylation. And I'll just show you again a result looking at 3 loci, where we're using an MCR-BC assay, but we've got bisulfite data as well, looking at the methylation of the target locus. So in wild-type plants, at these 3 loci, the level of methylation is high. If we look at a dicer mutant, the methylation is much lower at these 3 loci. This is in the root. So this is showing us that methylation at these loci is dependent on RNA. If we graft these dicer mutants onto a wild-type plant, what we now find is that the methylation level is restored. So this shows us then that the RNA that has moved from the shoot into the root is capable of directing the methylation at these 3 loci. And then this is another experiment, another control where we've used a polymerase 4 mutant in the shoot, and again the methylation levels goes down. So the mobile RNA is mobile as small RNA, as small interfering RNA, and at these loci it can do something when it gets to where it's going. Can it do transcriptional gene silencing? So a related question to methylation, because methylation is often associated with transcriptional gene silencing. And to address this, we were able to obtain some plants from Marjorie Matsky. So there are 2 lines in this experiment. One line has a gene called the target gene. So this is a GFP reporter with a promoter upstream of it containing a meristem-specific enhancer region. She's also included in these, she generated a silencer line. So this has a region corresponding to the meristem enhancer in the transcribed part of this gene. So this silencer will generate fold-back RNA that corresponds to the promoter of this target gene here. So when we've got 2 genotypes, target grafted to target, and we look in the roots, I hope you can see a few little bits of GFP, And that's because the GFP is brightly expressed in the roots of these TT genotype plants. If we graft on the TTSS genotype, so they contain both of these transgenes, we now lose the GFP fluorescence. And if I showed you the RT-PCRs, you'd see that you've also lost the accumulated RNA from the target locus. So you've got mobile silencing that we think is acting at the transcriptional level. And then corresponding to that, we've got the small RNAs and the methylation profiles that you would expect. So in TT roots, there are barely any small RNAs. In TT roots grafted to the TTSS genotype plants, you've got abundant small RNAs corresponding only to the promoter region of this gene and of all different size classes. If we use a DICER3 mutant coupled to the TTSS genotype, then we lose the 24s. Corresponding to that, in the control plant, TT, there's no methylation of this promoter. In this plant here, grafted onto this, there's high methylation at all three sequence contexts in the promoter region, and that is lost if the shoot is of the DICER3 genotype. So these mobile RNAs direct methylation to where they're going, and they can cause transcriptional gene silencing. Just a little side issue here. At this particular locus, it's clear that 21 nucleotide RNAs can move, whereas at the pi locus that I showed you previously, they couldn't. And so there's a bit of variation between all of this. So summarizing then what we know about mobile silencing, this shows us that 24 nucleotide and 21 nucleotide RNAs can move as siRNAs. Only the 24 nucleotide RNAs, as far as we can tell, has epigenetic silencing activity in these systems. An important point that I should have emphasized when I was describing the experiments with the Matsky system is that the silencing can be targeted and can initiate in the meristematic zones. That's where this promoter is being expressed that we used in those experiments. And this is important if you're thinking about a mobile silencing signal that might be transiently expressed and initiate a long-lived effect. In order for such an effect to operate, it needs to operate in meristems. And from these experiments, we can be confident that the effects are manifest in meristems. Final point, the movement is photosynthetic source to sink. In these experiments, for technical reasons, we've looked at movement from shoots to roots. But in other plants, with proper stems and so forth, then it's likely that one could get movement into the shoot apical meristem, the meristem that will give rise to flowers. And so one could think of these mobile signals, RNA signals, as initiating epigenetic effects that would be manifest in the shoot meristem. And in some instances, because we know that epigenetic marks in plants can persist from one generation to the next, one could think about transgenerational epigenetic marks initiated by these mobile signals. So this is where we are then with our understanding of mobile silencing. There's a bit of a problem, though, at least for Arabidopsis, because if we look at the mutants that can't make these 24 nucleotide small RNAs, the plants don't have a phenotype. They grow pretty well. Flowering is barely effective, the timing of flowering. The morphology, the stress responses, and we've looked very comprehensively at this, are quite normal. They're not hyper-susceptible to any stresses, unlike what it says in the literature. They're perfectly normal. So we haven't got a phenotype here, and this makes you start asking questions that whether or not you're looking at something that's really real. Well, we hope that we were. I mean, the system looks to be relatively sophisticated, and we thought it's likely that we are looking at a process that has biological relevance, and we thought we're probably looking in the wrong way. So Becky Mosher, when she was a postdoc in the lab, just did one of these sort of time course life cycle experiments. She said, when in plants are the 24 nucleotide small RNAs most abundant? And what she found was that actually they're present throughout the life cycle of the plant, but they're really highly abundant in the seed shortly after fertilization, a few days, 7 days or so post-fertilization. So then she followed up her analyses, and she showed that they were expressed at that time in the endosperm, or she thought it was the endosperm, and specifically from the maternal genome. So this then made us start thinking about the possibility that these small RNAs are not necessarily involved as signals of development or response to stresses or whatever, but maybe there's something to do with the interaction between genomes. And so we started thinking that they might have something to do with the parental genome conflict between maternal and paternal genomes. Or we thought maybe they have something to do with the compatibility between different genomes when you make hybrids, wide crosses, and so on and so forth. We haven't resolved this at present. We've been collaborating with Jeffrey Chen from Texas, and the data that he's generated is certainly consistent with the parental genome conflict idea. But, so this is an issue to be resolved. But what this made us start thinking, as I say, is of a role for these small RNAs in genome interactions. And we have tried to articulate our working hypothesis in these terms. That is that the transposon-derived small RNAs, or the 24-nucleotide small RNAs, what they're doing is they're allowing or helping a genome to define itself as self. If you like. So a genome will exist in equilibrium with the 24 nucleotide RNAs that it produces. But when you make a cross, when you hybridize one genome with another genome, the 24 nucleotide RNAs from one genome would find targets in the other genome that it doesn't have normally. And so you could envision that there would be hybrid-specific effects associated with silencing. So you've got small RNA from genotype A finding a target in genotype B. And this then could result in this target being silenced in the hybrid, whereas it wouldn't be silenced in either of the parents. One could also envision, because we know that small RNAs can initiate secondary small RNA production, that what could be happening is that you would have loci that give rise to secondary small RNAs that don't give rise to small RNAs in either of the parents. And these two could have silencing effects in cis or in trans. So, does this happen? Well, this is where we moved away from Arabidopsis to explore this possibility. We moved to tomato, and part of the reason for doing this is because in tomato you can make interspecific crosses. We figured that if you were going to look at this type of phenomenon, test this hypothesis, the wider the cross you make, the better it is. So we could make interspecific crosses in, between tomato and a wild relative. And we could also exploit a whole series of introgression lines that Dami Zamir and his colleagues had generated in Israel many years ago. So the crosses are between tomato and its wild relative Solanum pinelii. And the introgression lines, they've been through several generations of backcross, and they've got small regions of the pinelii genome in the background of tomato. So 8-3-1 has got this little terminal bit from pinelii, but the whole rest of the genome is from tomato. So a resource that one could use to look at these hypothetical effects. And the approach that we use to look at these hypothetical effects involve sampling small RNA from seedlings of the two parents in this cross. We looked at F1 and F2 hybrids that we generated ourselves, and we also looked, as I said, at various introgression lines. So this is part of the data, or these are part of the data that we generated in this type of experiment, and this is a control. So this is a locus that is expressed as small RNA in tomato, not in Solanum pinelii, at an intermediate level in the F1 hybrid. And in most of the introgression lines, this locus looked like tomato, and that's fine because this is tomato DNA at this locus in most of the lines. And then we had a few of the introgression lines where it looked like pinelii, and that was fine because it's got pinelii DNA at this particular locus in those lines. But we saw a number of exceptions to this, and here's one of them. So this is a heat map looking at the expression of small RNA from different loci. And there's one locus in particular that we have followed up on because all of the introgression lines produced abundant small RNAs from this locus, whereas neither of the parents did. So looking at this locus in a little more detail, it is a locus that corresponds to a protein coding gene. The small RNAs are derived from an intron, They're colored blue, so recalling the previous slides, these are 24 nucleotide small RNAs. And as I say, they're present in all of the introgression lines that we looked at. Now, we began to worry that, maybe these introgression lines, there was something genetically different about these lines. But what we found was that when we made our own F1s and F2s, that in 2 out of the 3 F2 lineages that we looked at, we started to get these small RNAs in exactly the same place. So we could recreate this. So it doesn't look as if there's a sort of genetic rearrangement associated with this. It looks as if it's an epigenetic effect. And linked to the notion that there's an epigenetic effect, when we looked at methylation at the DNA at these loci, so we found in the parents that these loci were essentially unmethylated. In the F1s, they were unmethylated, but in the F2s and in the introgression lines, the DNA at this locus was methylated, and the expression, so this is an RT-PCR assay, the expression of the messenger RNA from this locus was on in the parents in the F1 and off in the F2s and in the introgression lines. So what we've got here is, we call this a transgressive event because it transgresses the boundaries established by the parents. And it's a transgressive silencing effect that is associated with small RNA. This is another locus where we also saw a transgressive effect, but some of the molecular details are a little different. So this is the phenylalanine ammonia lyase locus in tomato, enzyme involved in phenylpropanoid pathway. And what we found was in one of our introgression lines, so this is 8-3, that there were highly abundant small RNAs from the PAL genes, whereas they weren't completely absent in the other lines, but they were present at a very low level. We looked at the alignment of these, and by using each, so PAL exists as a multi-gene family in tomato. There are unique sequence tags associated with each of the multi-gene family members, and we could infer from that, by aligning the small RNA sequences, that each of these PAL genes was converted into small RNA in this particular introgression line and not in any of the others. And looking at the alignment of these small RNAs with one of the PAL genes, one can see, first of all, that they're derived from an exon, not an intron. And secondly, I hope you can make it out, they're red and they're not blue. They're 21 and not 24 nucleotides. So these are most likely small RNAs associated with post-transcriptional silencing. I have to say that we don't know how it is, what initiates the production of these, and we're not being able to detect any epigenetic marks that are associated with this, and we can't rule out that associated with the hybridization event that there was some sort of genetic rearrangement that has led to the production of these small RNAs, but nevertheless, the production of the small RNAs is the manifestation of a transgressive effect on gene expression. And I could show you the RNA assays, but these are a little more colorful. These are the histochemical assays for lignin, so lignin dependent on the phenylpropanoid pathway, and in 8-3, the line with these transgressive small RNAs at PAL, the lignin in the vascular bundles is very low, whereas in all of the other introgression lines and in the parents was highly abundant. So transgressive PAL small RNA has got a transgressive silencing effect on gene expression and lignin production, and we're testing these lines now for other effects that you might think would be consequent to low PAL levels, so effects on disease resistance and so on and so forth. So just then to close by summarizing, so we think that there are many tens, probably hundreds of transgressive small RNA loci in these interspecific hybrids. They can affect gene expression, and consequently they can have an effect on the growth and development of hybrids. I've given you two examples here where it looks as if very different mechanisms are involved, and we think in other loci that we've looked at there may be still further mechanisms involved. So there's not just one simple mechanism behind all of this, but we do think that there's an epigenetic component to it. And then just to sort of link this with the first part of the talk, so what one might think of then is that there's the potential for plants to respond to various stimuli. So one could think of stress or genome shock in hybrids that initiates the production of 24 nucleotide small RNAs somewhere in the plant. These 24 nucleotide small RNAs can then signal through the symplastic movement mechanism to the meristem, where they could mediate the introduction of an epigenetic mark. That could then lead to a modified pattern of gene expression that would persist for as long as the epigenetic mark persisted. So in some instances, that would be for a time, and it would be erased at gametogenesis, or it's possible in some instances that the modified pattern of gene expression would persist between generations. And the transgressive effect that I described to you, the first locus, the locus we refer to as 806, don't know what the protein does, but that looks as if it's an induced epigenetic effect that is transgenerational, that persists between generations. So that then is, if you like, my summary, and just to acknowledge the people who've worked on these projects. So the mobile silencing project has been a fantastic joint effort by Attila and Charles, and Andrew, who helped out with the methylation analysis. The tomato work has been done by Shiva, and then Chris, Ruth, and Tom have helped with the bioinformatic analysis of the small RNA data sets. So thanks for your time, and I'll be happy to take questions.
Presume that there's no feedback loop to maintain silencing. So if you now remove the source of the small RNAs for, say, this cell mutant route, does silencing disappear?
Well, we're just doing that. We should have done those experiments before, and thank you for pointing that out. I know we should have done them. We think that once you've established an epigenetic mark associated with DNA methylation, that will persist, even in the absence of continued production of the signal. But you would only get persistence of C residues most likely in a CG context and not those in a C asymmetric C context.
Could you just comment on what the evidence is for the transport in the phloem and the symplasmic transport? I mean, I know that you don't get it into the guard cells, but that's an indirect evidence. And is there clear evidence that it's going symplasmically and that it's going through the phloem?
You're absolutely right. It's indirect evidence. So a long time ago, when Olivier first described the mobile silencing, he was able to show that if the GFP silencing signal moved into leaves, a GFP silencing signal moved into leaves in which the stomatal guard cells were symplastically isolated, then the GFP didn't get silenced in the guard cells. So indirect evidence, but I think pretty good evidence that it actually, at least into the guard cells, is symplastic movement. The other evidence, I mean, you're absolutely right, is indirect, but the kinetics of the long-distance movement of the signal, as far as we could tell, are pretty much the same as the kinetics of virus movement, and the pattern is also very similar to the pattern of virus movement. So consistent with movement through the phloem and through the symplasm. But I mean, you could, if you were being truly objective about all of this, say, well, all of this data from 50 years ago that was interpreted to indicate symplastic movement of viruses, maybe that needs to be re-looked at as well. But I mean, we do know that viruses move between plasmodesmata, so that's where we stand on that. But I'd be grateful for any suggestions as to how we could rule out apoplastic movement.
Well, if you get it into the seeds as well, I mean, you were indicating this, then wouldn't this potentially imply that there must be an additional mechanism?
Well, Rob Martinson has invoked movement of a silencing signal into the pollen sperm cells, and if that is happening, then almost certainly that would have to be apoplastic movement. I'd like to have a discussion with Rob as to whether or not that is actually happening. But anyway, that's another discussion to be had.